KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing significance of meiosis and new chapter, that is biomolecules. So biomolecules introduction in this video we will discuss, but so important so here even though it is a biomolecules introduction information, so few new bits and somewhat new information we can learn here. Okay, now significance of meiosis point of view. So significance means so importance. So what is the importance of this particular meiosis? So here what we have to do means here the meiosis is a mechanism by which conservation of specific chromosomes or chromosomal number of each species is achieved across the generations in sexual reproducing organisms, even though so up to here let us stop and let us learn the meaning. So now here you see that so meiosis is a mechanism. So meiosis process definitely is a mechanism. Why? Because there so very complex information and very depth information we have learned, isn't it? So there, so what is happening? The conservation by which conservation of a specific chromosomal number of each species. So definitely here we are conserving, or the meiosis process is a conserving of specific number or the specific chromosomal number. For example, so our parents are so having 23 pairs of chromosomes. So from our parents we are formed, isn't it? So means here when we are checking our chromosomal number, no, so even so we will be having so 23 pairs of chromosomes like our parents. So here our parents, so both so mummy and daddy having 23 pairs and 23 pairs, but in our body together 43 or 43 or 46 pairs are not there. So even we will be having, or we have 23 pairs of chromosomes only. Why? Because due to the process of meiosis or during gametogenesis simply, so what is happening? No, chromosomal number is reduced to half in both parents. So 23 pairs converted into 23, and there also 23 pairs of chromosomes converted into 23. So both together 23 pairs. So we are formed. So means we formed here. So likewise here what is the point here we can understand now. So this particular meiosis process definitely helps in conservation of a specific chromosomal number. Don't forget the important point of each species. Okay, next one here we have written somewhat information is achieved across the generations in sexually reproducing organisms. So here in all sexually reproducing organisms definitely, so this particular meiosis process helps in conservation of specific chromosomal number. Amma point purida? Right. Next one it is, even though the process of per se paradoxically results in reduction of chromosomal number by half. Once you see that, so thereby suppose in order to the formation of new offsprings by the process of sexual reproduction, definitely from the both parents so chromosomal number must be reduced to half. Then only in the next generation, no, so here in the next generation, no, constant number of chromosomes or conservation of a specific number of chromosomes here we can observe. So thereby here what is the point we are understanding? Meiosis we are calling by one more name reductional division, no. So thereby in reduction of chromosomal number by half or reduced to half. That's why in anaphase 1 we have seen we have drawn 2 chromosomes, even though we have taken meiocyte as 4 chromosomes. So what is the point? Even though the parental number or diploid number is 4, in after anaphase 1 we have drawn just 2 chromosomes in per cell or per cell, isn't it? So there, what is the point we are understanding now? After anaphase 1 or in anaphase 1 of meiosis 1, chromosomal number is reduced to half. So next one, it also increases the genetic variability. This is nalla remember pannikonga. So what is the importance of meiosis means that also helps for the what it is, bringing of genetic variability in the population of organisms from the generation to the next generation. So thereby here one point I said, no, 100% we come from our parents. So even though we have some kind of differences when compared to our parents, isn't it? Suppose here if both parents and offsprings are seems to be same, means we cannot differentiate. Like suppose if you see if you observe the China people, for our eyes point of view, so all China people will be like a same only. Means like that here also, suppose when we are observing among our family, so definitely we will have some kind of variations, or the differences will be there, height point of view, color point of view, stature point of view. So likewise definitely some kind of differences we will find out. So that kind of differences are coming because of the meiosis or because of the reductional division. Next one, variations are very important for the process of evolution. So don't forget, this particular point also is important. So variations. So what are the variations are coming in the meiosis? No, definitely because of these variations are base. These variations are base for what? So definitely important for the evolution. Okay, so in this particular point, so here the organisms are going to be evolved from the one generation to another generation, means that is only achieved because of the presence of or because of the mechanism of meiosis. Red point important. Chromosomal number is reduced to half. Next one, in organisms, if you see there, definitely brings the genetic variability. So that particular genetic variability brings the evolution. Simple only nalla save pannikonga. Next one, this somewhat information regarding meiosis or the significance of meiosis. So the lesson which gives the complete information of which one cell cycle and cell division chapter. In this particular information or in this chapter, so what are the points we learn? First of all, we learn the information of cell cycle, their phases of cell cycle, interphase, so G1 ES, G2 information we seen. Upper M phase mitosis point of view, so like a prophase, metaphase, anaphase, telophase we seen. Upper significance of mitosis we seen. Upper meiosis point of view, meiosis 1 and meiosis 2, like 2 types over there. And in meiosis 1 again karyokinesis and cytokinesis pathumbodu, ange karyokinesis, so prophase 1, metaphase 1, anaphase 1, telophase 1 we seen. Apro cytokinesis, apro again after cytokinesis somewhat gap we found, interkinesis we called adi. Apro meiosis 2 start achi, apro in meiosis 2 prophase 2, metaphase 2, anaphase 2, telophase mudinji. Apro now finally we have seen the significance of meiosis. So that particular information. Now we are going to start new topic, that is biomolecules. Actually I thought no, so when you will be here at the time I thought to start this particular topic, but so gradually these chapters are going to be finished, so that's why so I have started so this particular information or this particular chapter now. So either over a particular time definitely I have to give this video. Definitely I have to give this information to you. So let me continue to give like this. Okay, right. Now biomolecules introduction. So now here somewhat information my own I have given. So what is information? There is a wide diversity in living organisms in our biosphere. Suppose here now only we see wide diversity in living organisms. Means here when we are observing like organisms or the living organisms, definitely variations are there. Means tiger one type, lion one type, and monkey one type, or dog one type, or cat one type. Means here diversity is there. Wide diversity in living organisms in our biosphere. Okay, definitely differences we are finding, and definitely variations also we are finding. Now here all living organisms, here one more time once you see there, here all living organisms are made up of with same type of elements. So here one important point, sorry, we learn plants are there, animals are there, insects are there, amphibians are there. Likewise there are different types of animals, plants, so many types of living organisms are there. Ange all living organisms sorry eppadi sir form aichu means, so one point you see all living organisms are made up of with same type of elements. Same type of elements means chemistry nalli we will learn carbon, hydrogen, oxygen, nitrogen, phosphorus. So likewise there are different types of elements. So here chemical elements we can consider. So because of chemical analysis of what we are taking now here let us examine. Now we are learning all living organisms definitely the chemical composition is the same we are learning so let us get the information whether it is this particular statement is correct or not. Now let us see. So now because of chemical analysis of plants or the animal tissues and earth crust, analysis gives similar list. Same type of elements are present in all living organisms. Now that one we are comparing with non-living thing or non-living earth crust. Why? Because here once you see there, when we are observing plant tissues or animal tissues with earth crust koncham soil edthi test pannavanna, so there also what are the chemical elements are present in earth crust same chemical elements are present in plant tissue as well as animal tissue. So thereby here one point we are understanding, so no such differences could be made out. So definitely here we could not find out any kind of differences. We could not find out. So next one, all the elements present in the sample of earth crust are also present in the sample of living tissues. Correct only? Now that point only we have seen. So means what are the elements or the availability is there in the earth crust. So same kind of chemical composition we can observe in living tissue. One point only I am stressing more. Soil chemical analysis send panamana. So what is the chemical composition information varumo? So same list only here we will obtain for the or we will get for the even living tissues also. So definitely there is no any kind of differences we could not find. So here means there is no any kind of differences we can find. Okay, so now this point is important. Let us observe very clear information. So that is information only here I have written. However, a closer examination. So very like outer null view point of view, we have seen like this. So chemical analysis of both living organisms and non-living organisms, chemical analysis or the list will be same, a similar list, but once you see, however, a closer examination, study what is information we are studying, reveals that the relative abundance of carbon and hydrogen with respect to the other elements is higher in any living organism than the earth crust. So now here what is the point we are understanding? So generally all kind of chemicals composition will be same only in both living organisms as well as non-living organisms we see. But now closer examination point of view, what is the point we are this neat examination point of view important so that point once you see closer examination reveals that the relative abundance of carbon and hydrogen with respect to the other elements is higher in living organisms than earth crust. So thereby this particular point of view, what is the point we are learning? Hydrogen hydrogen முன்னாடி carbon hydrogen number will be more in all living organisms when compared to the earth crust. Point புரியுதா? one more time நல்லா சேவ் பண்ணிக்கோங்க. இப்போ first of all one point we learned, அது என்னன்னா, so chemical analysis of both living organisms and non-living organisms, no, there what is the thing, same kind of chemical analysis we will get, like that we learn. Ipo clear examination point of view, now we are updating our knowledge what it is, no, in comparison of both living and non-living organisms, no, in living organisms we are finding the relative abundance of carbon and hydrogen will be more when compared to the earth crust. அம்மா பாயிண்ட் புரியுதும்மா? கொஞ்சம் டிஃபிகல்ட் பாயிண்ட்டம்மா, கொஞ்சம் சேவ் பண்ணிக்கோங்க நல்லா. சரிம்மா, நெக்ஸ்ட் பாயிண்ட்டம்மா அது என்னென்னா, இது கொஞ்சம் ஈஸி தான். நெக்ஸ்ட் சைடுனா இது கொஞ்சம் ஈஸி தான், யூ கேன் அண்டர்ஸ்டாண்ட் வெரி கிளியர்லி. அது என்னென்னா, நவ் யூ see there. So how to analysis or how to get the one chemical analysis? Sir, how we can get the chemical analysis of a sample? How we can get? No, to get the chemical analysis, we can take any living tissue. So we can take any particular living tissue. So there the vegetative or the vegetables we can take, or a piece of liver we can take. So vegetables or the piece of liver eduthu. So now what we have to do, no, now we have to grind it. Grind. Ipo lab nalli paathumbodhu, if you see there, no, white color mortar and pestle we will see, no. So we will I will show you in our biology lab. So we have that one. So just name point of view try to get or grind it where. So in trichloroacetic acid, amma idu nalla save pannikonga. Ipo for getting of one chemical analysis of one living tissue, so what we are doing, we are grinding that particular what it is particular tissue in trichloroacetic acid. Appadena grind பண்ணும்போது what we have to do, no, while grinding, so this particular trichloroacetic acid we have to add. இப்போ trichloroacetic acid formula பாருங்க. So Cl3CCOOH, இது நல்லா சேவ் பண்ணிக்கோங்க. So important. That's why while grinding any particular living tissue, definitely if you would like to get the chemical analysis, means definitely we have to add the trichloroacetic acid. So now here, so using a mortar and pestle. So by taking mortar and pestle, by adding trichloroacetic acid, we have to grind the one particular living tissue. Then we can obtain the chemical analysis. Okay, man. Now when we are grinding, you know, definitely we will obtain the thick slurry. Definitely we will obtain the thick slurry. அதேமா சப்போஸ் மம்மி ஜூஸ் பண்ணும்போது thick slurry வரும், liquid எல்லாமே ஜூஸ் எல்லாமே, அப்புறம் so she will and juice she will give us. So likewise here also before filtration, so thick slurry we will obtain. Now what we have to do with that? No, if we were to strain this through a cheese cloth, this cloth name is cheese cloth, or we can use the cotton. So when we simply, so here what is the meaning of these lines, no? So what is the thick slurry is there, that one. Ipo best example I will give you. Ipo nalla save pannikonga. Ipo pathu, ipo mummy when she is preparing a tea, no, after preparation of tea she will filter, no, by taking off one filter, adu edo one name irukku. So by taking that filter, no, so she will filter the particular tea. Coffee means not required. Tea, why? Because their filter powder will be there, no, that filter we will not have. So that one we will separate, no. So likewise here also, when we are taking a cheese cloth or the cotton, no, we will obtain definitely two fractions. Idi ipo paathama, idi tea bowl. Suppose for example, so ange idi when we are using that particular filter, no, so like mesh like filter will be there, no. So when we are using that one, no, கொஞ்சம் டீ உள்ளே போகும், டீ உள்ளே போய் அப்புறம் மேலே அது டீ பவுடர் will be there, no, that one. Example, don't write a tea and tea இது டீ பவுடர் அந்த மாதிரி don't write. So examination example point of view. So when we are separating like that, no, so we will obtain two fractions. Two fractions and ரெண்டு parts ஆகும், two fractions. So biology point of view, two fractions, or the chemical biomolecules point of view, again two fractions are there. So one fraction we will be calling as a filtrate. Filtrate na what is the thing or what is the liquid ulle pochu, glass ulle or tumbler ulle pochu, adi we will be taking as filtrate, or the somewhat technically we are considering as acid soluble pool. புரியுதம்மா? acid soluble pool, once you see there acid soluble pool, so means while addition of acid no soluble ஆயிடுச்சு. ஏமா acid soluble pool or filtrate. And second one இதம்மா, second one இது மேலே இப்போ பாரு சின்ன சின்ன hair like structure, அது round appearing or not I don't know. So here மேலே thing no we will be taking as retentate. Once you say retentate or acid insoluble fraction. அம்மா ரெண்டு fractions அம்மா. One is acid soluble pool and one more is acid insoluble fraction. Or we can use another names. One name no filtrate and one more name retentate. amma point purida ரெண்டு fractions amma. That's why so here small rough diagram I have given to you. Once you see இது tumbler or ஏதோ ஒரு test tube, அங்கே what is the liquid present inside the container no, so that particular container or that particular liquid we will be taking as acid soluble pool. And what is the liquid or what is like waste thing is there no, or likewise only no, so that particular or what is the thing which is not inserted into the liquid no, that particular thing or which is separated we are calling as acid insoluble fraction or retentate. Now let us move forward. Now scientists have found thousands of organic compounds in the acid soluble pool. Amma pathika so from here I'm not konchi important. So scientists what they found thousands, not 1 2 3 4, thousands of organic compounds they found in the acid soluble pool ingema ulle. So what is a liquid which is entered into the container or there they found thousands of organic compounds in the acid soluble pool. Right. Now next one இது easy da, one or two times nalla save பண்ணிக்கோங்க. அது என்னன்னா all the carbonic compounds that we get from the living tissues. One more time. All the carbonic compounds that we get from the living tissues or the living organisms we can be taken as biomolecules. What are the carbonic compounds are there no? All these carbonic compounds, what are the carbonic compounds we are obtaining from the living tissues? Definitely these carbonic compounds we can consider as biomolecules. One lemma, simply once you see there carbonic compounds that we get from the living tissues. Thundos. So the carbonic compounds that we get from the living tissues we will be considering as biomolecules. Easy only no. Next one. However, living organisms have also living organisms have also got inorganic elements and compounds in them. So even not only these kind of carbonic compounds, so along with these kind of carbonic compounds we can find out the which one inorganic elements and some kind of compounds of them. Okay, next one. Chemical analysis. Sir, all the time only fresh materials, so suppose if not available what we have to do sir means, so chemical analysis we can obtain or we may obtain from the fresh weight. So fresh material, suppose if you would like to get the chemical analysis leaf or liver or either one particular plant material or the animal material we can use, or dry weight material or dry material or the dry material appadina. Ipo fresh material, ipo herbarium, do you know plant material, so suppose if you are keeping in a shadow places for a few days no, adi dry agun theriyuma. So there what is the differences between the fresh weight and the dry weight no? So dry weight nalli thanni irukadu avladha. So thanni will water when the water condition is lost no, that fresh weight no converted into dry weight. Next one, suppose if you burn, suppose if you put fire to that no any particular material, after some time no ash varum, white color, somewhat grey color, enna color, so ash varum. So that particular that one also which gives the what chemical analysis of chemical analysis of elements. So thereby here what is the point we are understanding no? So the chemical for obtaining of a chemical analysis, so here we can use fresh weight or the dry weight or the ash. So this is a somewhat introduction class regarding biomolecules. Actually even though it is introduction class no, very important. So here, so what are the points here we can say means, So while grinding the particular material for a chemical composition point of view, here we will add the trichloroacetic acid. So thereby we will obtain the true fractions. One is acid soluble pool and one more is acid insoluble fraction. Acid soluble pool we are calling by filtrate, and acid insoluble fraction we are calling by one more name, retentate. So this is somewhat information regarding biomolecules. In the next video I will come with the continuation topic of this. Thank you and be safe at home.